Uh, Thanks for staying until the end. So we have our last lecture, and it's my pleasure to have Dennis Wang here, who is have joined positions at Imperial and A Star. Um, so he is one of my closest collaborators. I'm very happy to have you, Dennis, here. Thank you.
Great, thanks, Mauricio and Magnus, for inviting me to this summer school. So, yeah, I'll try to be as light in this talk as possible because I know it's been a long day, and I've kind of changed my aim for this talk. Instead of giving something kind of deep into a topic, I'm going to go broad because it's kind of end of the summer school, and I kind of want to try to maybe summarize the best I could from at least what I learned today. So I'm going to talk more about drug development rather kind of just broadly across the whole drug development pipeline rather than just specifically on drug discovery. And one reason actually is drug discovery. There's been already quite a large number of compounds already found for even particular targets or diseases. So really the challenge in terms of drug development is how do we actually narrow down the large portfolio of compounds that, say, a pharmaceutical company might have into one particular one that will be approved to be treated in patients. Okay. Let me actually just get a quick survey. How many people work on kind of data relating to drugs? I know our previous speaker have. Okay, a few. Okay, great. So I'll maybe give more of an introduction on kind of the whole drug development process. So there's quite a lot of opportunities to work in this field, and I think over the past 20 years, pharmaceutical companies actually have become more open towards collaboration in terms of like with academia in terms of doing experimentation and also data analysis. So this is a website for my old company. Oh, I actually forgot. This is actually quite important. Just my disclosures of my previous funding and projects with some pharmaceutical and biotech companies. So it's just a kind of a disclosure. My kind of views are maybe colored by those projects as well, and they're also my personal views. And so yeah, one of the companies is AstraZeneca, and then they have like an open innovation portal where they actually enable researchers to apply to access their compounds, either to kind of use the compounds to for experimentation, or they may even have data from, say, failed testing of those compounds that they can also share with you for kind of analysis and modeling. And a company like AstraZeneca will have 4 to 5 million different compounds, and probably thousands that they have tested. Some that have been successful, made it to patients in the clinic. Most were not unsuccessful, and so they have actually a lot of data just sitting around in the companies. So the drug development process is quite long, tedious, and costly. Okay? So it starts with TI, which is target identification, and it ends with kind of a success of a phase 3 clinical trial. Okay, that gives evidence to a regulator like FDA to approve the drug. Right. As you can see, that the total time can be anywhere between like 9 to 15 years. Okay, traditionally. Of course, there are faster ones, like for instance, some of the therapeutics relating to COVID, right? But there could be longer ones like Alzheimer's disease, right? Which has been a very difficult disease to treat. And the total cost can range from like $500,000 to billions, right? Because the cost actually encompasses all the failures. So along this stage, right, you remember the previous graph I had, you have 10,000 compounds, right? So some of it will make it through to each of the following stages, but most will fail, right? So testing those compounds, the failures cost time and money, right? So really what we want to do with kind of data analysis, the goal of our data analysis and modeling is to try to reduce that time and cost, right? To kind of speed up this process, but also kind of reduce the chance of failures, essentially, because the failures cost a lot of money. So this is the approach I'm going to suggest is kind of based on some review of data by pharmaceutical companies. So this is a publication almost 10 years ago by AstraZeneca looking at their successful and failed trials. Okay, and then so their successful and failed projects and trials. So basically, the first panel, A, shows that you're more likely to have a successful drug pass phase 2 trial. So phase 2 is a phase in human testing. If there is general confidence in their science teams around how they select patients for that trial. Okay, and then in panel B, they also look at it in phase 2, successful phase 2 trials. There's more successes when they ask kind of their scientists about the genetic linkage of the disease. So that their scientists are confident that there's a link between the target, right, maybe the gene target of the drug and the disease. That there's actually a mechanism there, right? Like for instance, genetic associations. And then for kind of phase 2A, kind of a slightly earlier phase of drugs where they assess general efficacy and kind of safety of drugs, they found that kind of if they have some biomarkers, so genes that may not be targets of the drug, but maybe just any kind of features that may be predictive or associated with safety or efficacy or potency of the drug, that also increases the chance of success in the clinical trials. Okay. So this is what those are kind of the 3 areas I will focus on today, because there's a lot of areas, examples and case studies along the drug development pipeline, but I'll try to give some examples in terms of the first stage, kind of identifying that linkage between the target and the disease, right? And I also talk about how we kind of design, how we use kind of machine learning methods to design the drugs to better target the genes. And then secondly, I'll describe a couple of examples where ML can be used to predict the efficacy of a drug, right? And also identify biomarkers. So this is done in kind of the preclinical stage of drug development. This is where you still have a lot of drugs, right? But you're not sure, which drugs you should take on to really test on patients, right? Because you do have to be sure, even though they're clinical trials, and people kind of volunteer to be on these trials, you don't want to give them something that will harm them, right? So there is a lot of kind of prediction and analysis done preclinically on what's known as preclinical models. And then on the third stage, where you're actually testing the drugs on in 3 different clinical trials on patients, I'm going to describe how we can improve patient selection and also try to predict possible side effects in patients. Okay. So identifying the target. So the kind of the most standard way, I guess, in the genomics era is people have conducted kind of genome-wide association studies. Take a large population of diseases and diseased individuals and healthy individuals, and try to find genetic variants that are associated with disease, right? So how many of you have seen this kind of plot? Because I noticed I say, okay, Manhattan, okay, good. I don't have to describe Manhattan plot. So obviously, yeah, the vertical axis is kind of an association p-value of a single genetic variance associated with disease. I'll just highlight kind of a gap right now in this analysis is that the genetic variants here have been focused on variants that are in genes, right? But actually 99% of the genome, of the DNA are actually in regions where there are no genes, okay? So actually a lot of variants that may be significant, we can't actually map to a gene. So then you ask, okay, what do you target there? Okay. So that one I don't have any solutions for it. It's like a currently ongoing problem. Okay, and throughout this talk, I'm going to probably mention more opportunities than actual solutions to maybe try to motivate you to go into certain areas. And so the outcome of what they want to see from these genetic association studies is basically if those genetic variants or combinations of genetic variants, they call it kind of polygenic risks, may be associated with disease. Okay, so I remember earlier there was a little bit of discussion on kind of the effect of ethnicity and ancestry around targets. So this is an analysis done by my lab in Singapore, where we trained a polygenic classifier on Singaporean cohort that's mainly made out of people with Chinese and Indian ancestry. And then we test on UK cohort, UK biobank, for instance. And I didn't show the negative result here, but we predict reasonably well on UK biobank Asians, which are predominantly Indian, from Indian ancestry, and some East Asian, but not many, right? The prediction is not as good as our ATTRACT cohort, which is a Singaporean cohort of similar age, okay? And then what I'm not showing here is the Caucasian British cohort, which is basically you don't see any effect. But one thing to highlight, you'll notice the confidence intervals are very large here, right? It's because of the sample size, where UK Biobank under recruits because people who volunteer are generally not ethnic minorities, right? So I think we can really strengthen that evidence by recruiting more from some ethnic minorities, but also to highlight that comparing the Singapore cohort with UK Biobank is that there are obviously also some environmental effects, right? Just because you're an Asian, similar ancestry doesn't mean you have the same risk factors either. Okay. So that's kind of more yeah. Feel free to interrupt me. Yeah.
So you said that the fact that the scores are not slightly different between similar ancestry is because of the environment. That's what you said. But
Partially, yeah.
can you prove it? Why is that the case? Because how can we tell that it's the environment?
Yeah, obviously. So I'm in this analysis, I'm kind of like controlling for ethnicity here, right? So with the genes, and also I'm also controlling for age, right? So kind of by just exclusion, right? What is kind of variable here is kind of environment. So it's if you're controlling for say one ethnic group, but in two countries. So this is in Singapore and UK. Even though there are Indians living in Singapore, Indians living in UK, their ancestry, they're relatively recent, people who recently migrated, like maybe 2-3 generations. So we don't think there will be any genetic drift. So kind of just what's left may be just due to environment. We obviously environment, I'm using the term environment as a very broad term, right? It can be anything from like behavioral the food they eat, right? So the diseases here are cardiovascular metabolic diseases, right? So kind of air quality, diet, these will all influence. So I term that as kind of environmental factors. But there's quite a lot of factors, right? That's kind of going to be a height. Yeah, exactly. One difficult challenge is very difficult to quantify those factors. For instance, diet is actually very difficult to quantify. You give the people surveys, they often lie on those surveys. And so what is kind of a, I think I think we even talked to Microsoft or like Apple about like maybe VR headsets that actually will capture the food they eat. but yeah, that's that may be future work. Anyway, so that's kind of just a quick like overview on like a population approach to identify possible targets. I'd say most of actually the really kind of personalized drugs have been based not on kind of population, not so much population data, but more on cellular data. So really kind of deep biology. For instance, in cancer, they've done a lot of biology, just understanding the cellular pathways of cancer cells, right? Different types of cancers. And this was like there's a seminal paper over 10 years back on like the hallmark processes in cancer. So a lot of the drug companies have been focusing on these particular pathways to find new targets, right? And around kind of after publication of those hallmarks, I was working with Microsoft Research, and we had like a PhD student there try to identify one of the hallmarks of hallmark pathways of acute myeloid leukemia. So we built basically a network model of an acute myeloid leukemia cell. That's this, and it's a very tedious process of the student like reading a lot of papers, looking at a lot of experiments, and initially actually building an ODE model for this network. Then we kind of simplified it into a more of a Boolean network for better interpretation. And the point of building a network is that you can perturb the cell to determine two states. Sorry, it's too small. One state is obviously proliferation. For cancer, proliferation is bad. Apoptosis or death of the cell is good, right? So basically, we're trying to build a network model of a single cell that, based off of basic experiments and literature, to predict one of two states, right? And then we can fit that model using parameterize that model using existing data, and then predict, simulate that model and predict the dynamics of these individual proteins in the network model and the cell fate in different drug conditions. Okay, so this is just example showing the predicted cell state here for each of the proteins, and then there was experimentation to measure the actual protein expression level of the cell in these states, right? So this is a very low throughput approach, right? Because it requires a lot of kind of manual labor and deep understanding of the pathways, right? People have now proposed more higher throughput approaches using kind of the large single cell data sets that Mo described earlier. And instead of kind of reading papers to parameterize these ODE models, using machine learning to optimize on this loss function between the predictions from the ODE model and the observed experimental response to predict the parameters of the ODE model, right? So that's not something new. People have proposed using machine learning to predict parameters of ODE models, but this one, I think, from Chris Sanders' lab, Cellbox, I guess, is applying it to this kind of application of cellular network modeling. Okay, and this example, I think Mo gave an example, excellent example of his work of now there's a lot of single cell data, right? So you can actually infer perturbations. So how one cell may change, one cell state characterized by its expression profile may change into another cell state, right? And then this one, they propose like kind of more mechanistic approach where they look at the expression profiles of each individual cells and build kind of co-expression networks. So this is basically building a gene regulatory network for each cell type, right? And then showing that perturbations between from one cell type to another cell type can be described by a gene regulatory network. Okay? So it's just a more kind of mechanistic way of describing cell perturbations. Okay? But I think this is quite exciting because the abundance of single cell data will obviously give more information to how we determine how will give us more kind of mechanistic understanding of how cells may respond to different perturbations like drug perturbations or other kind of perturbations. So just quickly wrapping up this section here, I just want to highlight some of the, I guess, the challenges with data analysis in terms of, using cellular approaches for identifying new targets. One is with single cell data, I sometimes look at the single cell t-SNE plots. They look almost like kind of jelly bean jars a bit. And it's like, you can cluster and segment all you want, right? But it's like, how do you know how many are actual real cell types, right? How do you determine that number of cell types, right? So that's still an open question. And another thing is that a lot of this data is still really just at one time points, and it's just, we can do associations with, single cell types with disease outcomes, but we don't know if that's actually causal, right? That might be actually a result of the disease, right? That gene expression or cell state might be a result of the disease rather than actually driving the disease. Another thing to consider is actually most of the resources, single cell atlases here, and there's more and more of them, which is great. They're basically covering all the different type of organs and tissues that you have in a human, right? But most of them are, through donations, mostly are from healthy donors. So it is slightly biased towards kind of, if you're studying kind of diseases and drug treatment, you should be aware that, some of the data, some of the, say, gene regulatory networks that you build may not be representative of actual disease states, okay? And then fourth problem is actually, when you take the cells out, a lot of it, when you donate the tissue and take the cells out, they're actually dying, right? So actually, the cells, there's kind of it's been found that there's actually specific gene expression changes and processes that get activated when tissue and cells die. So there's kind of a post-mortem effect in the cells, right? So, but a lot of what we're trying to infer is when the tissue is kind of alive in a person, right? So that's also to keep in mind. And we've already touched upon kind of environmental and ancestry effects. There is so the Singapore is also part of the Human Cell Atlas. There's some unpublished data for kind of the Asian Human Cell Atlas that there's actually differences in single cell gene expression between Asian ethnicities, which you actually don't see in the bulk tissue, but you do see some differences in the clusters from the single cells. Not sure if that's real. Again, like going to my number one point, but yeah, there's maybe some evidence around kind of environmental ancestry effects. Okay. Any further questions around this section before I move on? Okay. All right. Yeah, feel free to interrupt as well. So just kind of going to the next section. So after you determine your target, we need to kind of identify a drug, right? Identify drugs that can inhibit or affect that gene target, right? So this kind of blurry pictures from a publication over 20 years ago, where kind of traditional approach basically starts with a medicinal chemist, right? Determining what we want from a compound, right? And just really being brilliant. I'm not sure if you guys are fans of Breaking Bad. I am. You know, you get like a Walter White character that can think of a very potent chemical compound, and then you know they can you do some preclinical experiments to measure the effect of that compound, and you know, fingers crossed. And they've done studies that, you know, really good chemists can consider multiple factors, right? So what are some of the factors to for a good compound or a lead compound, they call it? So you want to kind of optimize on one, like potency. So really it hits the target and the drug affects the target, right? Bioavailability, that there's, you know, abundance of that drug, right? It doesn't get consumed somewhere else. That there is actually that drug in the tissue that's diseased tissue, right? ADME is kind of, I'll describe a little bit later, but it's kind of absorption of the drug by the body. And then, and also it should be specific to that biological target. One problem in drug development is kind of off-target effects, that drugs might target something that's actually critical to your normal bodily function, right? So that's one of the reasons why you have side effects. You want to also decrease time of chemical synthesis. That's really important for a pharmaceutical company who spend a lot of time and effort trying to scale up manufacturing of the drug, right? If you want to make a drug for the masses and make a lot of money, you have to also decrease synthesis. And also you want to decrease toxicity as well. So this is kind of like, you know, what you want, right? The kind of the ideal case. Right. What often happens is like people often tune towards potency of the drug target, but what we see is like with potency of drug target, you often get more toxicity as well. And this is kind of more of the reality for most drugs, right? You often get some toxicity, some potency, and some bioavailability. So yeah, you have to be realistic. But this process of kind of screening for drugs has been sped up. Okay, so one kind of advent is like you know, the use of transformer models, right, in AlphaFold to predict 3D drug conformations. Why this is important is because then you can predict where a drug can potentially bind, right? So you can actually screen compounds for what drugs bind to a particular fold in a protein, right? And then, you know, I think this was like kind of a press release that from University of Toronto, that they were able to use AlphaFold to find a binding for a CDK protein important in liver cancer, and then they were also able to synthesize a new compound for binding that target. Obviously, new compounds are not like completely new. So people have been using, actually, the field of cheminformatics have been using machine learning models quite a while now to do de novo synthesis by training models on known chemical compounds and what's been known to kind of bind specific protein targets, right? So there's kind of a whole, you know, class of models now. I'm not extremely read up on what's the best here, and I think what's the best here will depend on a number of factors that I've listed here, right? And then I think the challenge is really to how do we optimize to the various kind of qualities that we want from a drug. And because this field has been like expanding rapidly. There's been a lot of toolkits, models being developed. And so there's also been some work in terms of building kind of toolkits. How do we benchmark these models, right? So this is from an initiative started in Boston, try to build kind of a therapeutics data commons. There's been a lot of kind of genomic data commons around. So they're kind of using a similar principle of building data commons for therapies where there's standard, there's chemical compounds, bioavailability assay, drug response assays that enable people to kind of test their machine learning models using these standard data sets, right? And be able to kind of benchmark how their models do in terms of predicting drug target binding or absorption or toxicity and so forth. So some of the main challenges I think with drug identification, especially kind of trying to predict the activity of the drug on the target, is that a lot of these models are doing the modeling as if the target molecule and the drug are in isolation, right? Usually in like some aqueous solution void of any other proteins, right? So, but one thing to remember that actually that the targets often exist in complexes. So they're actually, the target protein might actually be binding other molecules, right? And that could actually change the conformation of the protein, right? Again, in different diseases, different people, the protein might have a different conformation, right? And similarly, mutations also affect the conformation. Environmental factors may also affect conformation of molecules. But there are now tools, using some of the AlphaFold predictions to predict also the effect of mutations on wild-type proteins. And then similarly for the drug, I mentioned that off-target effects is one of the kind of the biggest enemies towards drug development, because drugs will also bind to other molecules, right? And so that makes the kind of the search space quite large, right? You not only have to kind of model this particular drug with this target protein, but you also have to model all the other possible proteins that might exist in the human body. Almost. Okay, so that's mostly done kind of in silico. Now, before you can go on to patients, you do need to still test the drugs in actually organisms, right? So you can test them in the lab on specific cells or organs or even animal models, right? And the main kind of output you're looking at here in preclinical testing is a dose response. Okay, so the response of the cell as you increase the dose of the drug, right? So for something like in cancer, we're trying to kill the cell, right? The cell viability, the number of viable cells will go down as you increase the dose, right? And then pharmacologists pay attention to kind of the IC50 metric, so the concentration where you get kind of 50% inhibition effect. Okay, so but this dose response curve, you can imagine there's actually many different metrics you can get from this type of data, right? And what generally the kind of the main readout here is you want to identify whether that drug in that experiment has a response, whereby basically the IC50 the cell viability decreases, right, with a little bit of the drug, or non-response, where doesn't matter, you could give a very large concentration of the drug and you don't see much of a response, right? So one problem with giving a large concentration of the drug, right, is because pretty much any compound you give to a person, it'll become toxic at some point, right? Like even water, right? You know, if you drink too much water, it can actually even kill you as well. So therefore, you want a kind of a response with just as little of the drug as possible. So that's what we're trying to optimize on. My work has been mainly focusing on what are the predictors of response and non-response using more genomic or genetic data. So there's been some kind of some data sets generated to help with that analysis. One is from the Sanger Institute, where they profiled a lot of different cancer cell lines, almost 1,000 different cancer cell lines across several hundred different drugs that's been kind of shared or no longer under patent. And then, you know, they basically collect a lot of dose response curves, and then they also collect genomic data from the cell lines, right? And basically, we're trying to use the genomic features to predict this dose response, right? So this is just showing you kind of the general size of that data in the context of the kind of the hallmarks of cancer I showed you earlier. So that's how they kind of decide actually on the compounds that they're gonna screen, is basically on those previous experiments, cellular experiments on kind of hallmarks of cancer. This is one example where we earlier analysis where we just looked at kind of that drug screening data and found that basically there's a lot of heterogeneity, right? So this is just two drugs that are BRAF and a BRAF inhibitor and PIK3 inhibitor that's targeting that has been tested for melanoma, okay? And then, but this is just showing even within cell lines, just like patients, there's a lot of kind of heterogeneity, right? And it's really hard to actually draw the line of where you think there's actually response and non-response. So this is where it's a tough problem. So it's easier to just pose it to the community as kind of like a Kaggle-like challenge. I think Louisa mentioned, you know, a similar challenge she did with, sorry, not Louisa, Gabriella, right? Gabrielle on kind of predicting DNA methylation. Similarly, we divided the drug response data into training and test set, right? And so this training and test set could be divided by drugs or by cell lines, right? And then we pose to the community, try to predict dose response, mainly IC50 values, or in the case of drug combinations, it'll be a thing called a synergy score, which is like kind of a three-dimensional dose response. And then we benchmark those predictions, right? So so what's here is the results of one earlier challenge we ran, where there were 76 submissions, so 76 teams who submitted prediction results for drug combination prediction. And we benchmarked the predictions using kind of random model, and then and also for kind of a upper bound is actually just replicates, the correlation between replicate experiments. Right? So if you were to repeat that dose response experiment, what would be the correlation between the repeat experiments, right? So you see kind of a spread, and actually quite a lot of algorithms actually predict quite well on the level of replicates. But in terms of absolute scale here, the Pearson correlation is still not very high, even for replicates. And then another observation we made is that no matter kind of the type of model or approach they use to kind of model to describe the relationship between genomics and drug response, there's a good number of drugs that are predicted well, but also still a large proportion of drugs that are just consistently predicted poorly. Okay? And we kind of want to understand a bit why that was the case. And we dug a little bit deeper into the dose response data, and then basically found that the dose response data, so the dose response variable, what we were trying to predict, is actually quite variable, okay? Even across replicates, right? And so there's actually quite a lot of uncertainty in the dose response in IC50s. And actually all the models weren't taking into consideration of that uncertainty, not because they didn't think about it probably, but also we didn't there isn't actually many replicate experiments. So you couldn't actually kind of estimate that uncertainty, right? So we got some more replicate experiments, and then we found that there's basically what's happening with the IC50s is that there's more uncertainty when you go beyond the dose responses that were tested. So what the current algorithm is doing were when you run the preclinical test, you have to kind of make a best guess at what doses to test, right? Sometimes if you guess too low, the actual IC50 value will be actually beyond what you actually tested, right? So you may have to extrapolate that curve out beyond this dotted line, which is the max dose concentration. Obviously, when you extrapolate the concentration, the uncertainty is larger, right? So we quantify this uncertainty by fitting a lot of GPs to as dose responses, and the confidence interval as estimated by GPs show that basically you get much larger uncertainty outside the dose response range compared to inside. Very logical. It's not groundbreaking, but it's something obvious that I think a lot of people didn't consider when looking at this type of data. Okay. Another thing I think people don't consider so much, they kind of generally focus on IC50, but this is actually a curve, and it's a best fit curve of actual specific responses. So the experiments didn't actually test every dose possible, right? Because actually doing preclinical testing is still relatively costly. So you get, measures, and then you fit a curve, and then most of people just use the IC50 value, but there are some other metrics you can get from the curve, like AUCs or the Emax. So the curves, a lot of curves actually plateau with increasing concentration because some cells may have actually resistance to some drugs, right? So this work, now we're looking into how do we, instead of predicting those summary statistics like IC50, can we actually predict the actual dose, the specific individual dose responses? So this is work by Juan Jose, who's actually here today, where we fit multi-output GPs to model the distribution of responses for each dose. Okay. And another thing that kind of plagues preclinical drug screening is actually reproducibility of predictions. So in the data set we looked at, there's actually been two versions. So there's a newer version called GDSC2 that has since been generated. And then and this right figure shows basically models that were trained on GDSC1, predicted on GDSC2, and vice versa. Right, and it basically shows the blue lines are our models, and then the yellow lines are actually the observed data. Okay, and it basically shows that yes, you can make predictions, which is good when you train on one data set and try to predict on another. But there are definitely cases like this where there's actually it's still not very reproducible. These experiments, basically, right, and then. And we can't really predict what can't be reproduced. Okay, so those are some of the challenges. There's some other challenges with testing on preclinical data. One thing I didn't touch upon, most of our data came from what's known as like 2D cultures, basically kind of cells taken from a patient, cultured in a petri dish laboratories. There's also a lot of animal testing that goes on for drugs, and that's to also look at the factors of pharmacokinetics, which is kind of how the body absorbs the drug, right? But obviously it's quite obvious to tell that actually the physiology of these models are very different from humans, right? A mouse is very different from human. Their liver is gonna, function a slightly different way, right? And then with 2D cultures, there's not even livers to kind of metabolize the drug, right? And then obviously I said reproducibility is still an issue. When we go learn models on 2D cultures and try to predict on mouse models, animal models, not everything validates. Okay? And that's not because we're particularly overtraining. It's actually because the models are fundamentally different, and it's just really hard to reproduce these dose response curves. Okay. Everyone still with me so far? Okay. All right. Good. Okay. Last bit here, I'm going to move into kind of clinical testing. So now, if you're successful with your preclinical testing on cell lines or animals, then you have to test on humans, right? And then I guess another survey: how many people work on kind of have worked on clinical trial data or drug testing? Oh, okay, a few. Great. So this is the most costly. This is where all the money is actually, right? So it's actually if you want to get a job, get paid. This is actually where the highest potential to find a job, because this is the most costly step of the drug development process, and it's costly mainly because of not actually administering the drug, but actually the patients, getting the patients. All right, so pharma companies, when they sponsor a drug trial, they have to pay hospitals a lot of money to recruit patients. Okay, and then take measures of the patients. Obviously, with the NHS stretched, right? Doctors, nurses' times are very valuable, right? That's the case everywhere in the world. So it's really important to make sure we actually recruit the right patients into the trial, right? And what you want to do is essentially for drug trial, the main readout is a survival curve, right? So you look at, say, the survival of the patient with the drug and compared to without the drug. So this is actual data they're testing a combination. So they're testing this therapy with a placebo compared to this therapy with another drug, actual drug, right? And what you want to see is basically you want to basically recruit patients where you will likely see a difference, right? So you want to recruit patients that would benefit from that particular drug. Okay. So even before considering any drug treatments, what's actually difficult with this problem is that even the diseases, whether a person is a patient or not, is actually very difficult is a very difficult problem, right? So even classifying whether a patient should be eligible for this drug is a challenge. And this is a problem I work on in a disease called pulmonary hypertension. It's a disease where it's in between a heart disease and a lung disease, and that's actually its clinical classification. So it doesn't have any actually like pathological classification. It's a classification by exclusion. It's not particularly a problem of your heart, and it's not particularly a problem with your lungs. Okay, and people die from it because they have a heart attack. Okay, and to get diagnosed, it's a really windy path because it's clinical classification by exclusion, essentially. Right? So we ran some early trials of these pulmonary hypertension patients, and then so one thing we asked, like, if the clinical classification for those patients are quite vague, right? Can we use molecular markers? So we basically took blood from these patients and profiled their RNA in blood and looked at pulmonary hypertension patients that are longer-term survivors or non-survivors, and also healthy volunteers, right? And essentially, there's kind of there's still quite a bit of heterogeneity there, right? So even essentially even molecular markers are not very distinct for this for these patients, right? So then how do you really classify those patients? So we're taking essentially approaches used in like single cell. So instead of clustering cells, we try to cluster the patients, right, based off of their RNA profiles, right? And then, so this is work developed by a PhD student of mine, Socrates. Well, he's graduate now, and he's actually working on single cell data now, but kind of developed a pipeline mainly to estimate the number of clusters, okay, by looking at kind of cluster stability metrics. So it's a little bit of a Monte Carlo method where we just go through all the different clustering parameters and take kind of 14 different measures of cluster stability to determine what clustering parameters and also what input values to use. So whether you use the whole transcriptome or specific genes, okay? And long story short, for pulmonary hypertension, we discovered 5 classes, new molecular classes of this disease, okay? In the cohort, 2 of the classes were quite small, so we couldn't do really any further downstream analysis. But 3 of the classes, they differed in terms of survival of the patients, okay? And then it's really important for these kind of unsupervised learning studies that you also validate in an independent cohort, right? So then we took it to a completely separate cohort to classify the individuals into those classes, and we still see differences in survival. And then we also looked at what are the kind of standard clinical measures that are just routinely collected for those patients and see how they might differ depending on our classes. So some of it, actually quite a few of the clinical measures did not differentiate between the classes, right? Some did, which is sort of good, because actually if the clinical class measures actually did differ between the clusters, then you don't really need a molecular marker. So it's kind of showing that there's some kind of molecular classes of patients that can't be characterized by the routine clinical measures, right? But they actually differ in terms of their prognosis. Okay, so basically in terms of, just in terms of like actually going for a trial, then you may want to recruit, particularly from those classes, right? You may not want to recruit for, say, introducing a new drug. You may not want to recruit from classes 1 and 5 here, right? Because they're already going to be like kind of long-term survivors, right? You may want to recruit from class 1 who may actually benefit more from the drug. Okay, all right, that makes sense. Okay, good. So another area that people can work on in terms of data analysis is predicting drug side effects. Okay, so this is usually tested in a phase 1 trial, kind of the first stage of the clinical trial, but that's with a very small number of patients. Okay, and then even after a drug is approved and out in the market, they still measure side effects, right? So there's been several landmark cases of pharma companies having to withdraw their drug after it's been on the market because they later on found some side effects, and it cost the company a lot of money Not just with loss in sales, but they actually can get lawsuits from people who actually got the drug, right? So it can be quite costly. So what a lot of pharmacologists have done is try to monitor, go through clinical records and compare drug-drug interactions. Okay. So one thing I think I haven't mentioned is that most patients aren't just taking one drug, right? If you are a sick individual, you're going to be taking multiple drugs, right, to help with different symptoms, right? And then there's also other things you're injecting into your body, like food and supplements, right? So there's actually while in a clinical trial setting, you might be very controlled, right? Those patients may not be taking certain drugs, but when you're going out into the market, people are going to be doing different things, right? Doing recreational drugs, doing pharmaceutical drugs, whatever, right? So there's a lot of monitoring from looking at clinical records to see, what drugs this patient might have been on before, what new drug have they taken, were there any possible side effects? And then University of Liverpool, for particular HIV, they kind of created a database describing all possible drug-drug interactions between HIV drugs. Okay? HIV is a case where they take, HIV patients take actually a cocktail of drugs. They actually take many drugs, actually. Some to manage symptoms and some to directly regulate the kind of T cells in the body. Okay? And so they've kind of been doing monitoring of kind of drug-drug interactions. And a PhD student there, I could supervise Tao, has been basically learning from, so this is kind of actually human-generated data, drug-drug interaction classes, okay? And basically, the pharmacologists have, created 4 different levels of drug-drug interaction severity, right? You can fit a convolutional neural network to predict drug-drug interaction classes. Okay. Interestingly, not many people have worked on this type of data. Right? Anyone work on drug-drug interactions here? Or okay, yeah, it's interesting. There's actually a lot of these like well-curated databases of drug-drug interactions that's existing, but actually very few people work on them. Most people work on drug potency, like effect, but not really side effects. But actually, side effects probably cost more money than actually beneficial effects. Okay, I'm gonna just last few. I still have 30 minutes. Or? Yeah. Okay, good. Last few slides, I'm gonna go through some of the challenges with clinical data. One is missingness, right? Obviously, like just like omics data, clinical data probably there's even more missingness. This is on the left here. This is actually like real data that we had from the pulmonary hypertension patients. I gave the example earlier, right? So this is kind of the black is kind of the missing data we have. We use multiple imputation to impute these missing values. But there's some more developments on using deep autoencoders to actually predict missing values in electronic health records. This particular paper, this particular method used basically a deep autoencoder whereby you iteratively drop out input variables to train a model, right, to predict kind of missing values in the output layer. And so this is quite critical. So missing values is a big problem with clinical data. Another problem is basically unstructured, how unstructured clinical data is. So most of the clinical data are actually in sometimes even handwritten notes by clinicians, but now they're digitalized, but it's really just kind of a free text a lot of times. Even when it's not free text, the terms they use are not standardized. So I think for something like heart attack, there's actually 50 different commonly used terms by clinicians to describe a heart attack, right? So there's a lot of work done by kind of data science community to make some standard terms. So some of the standard terms or vocabularies are like ICD codes, SNOMED codes, and then there's OMOP terms. And then, so that's where you want to store those standardized structured data into data warehouses for analysis. But this process of converting unstructured clinical data is still very manual. Right now, we basically have like data curation teams of several dozen people just talking to doctors, trying to understand the clinical notes. Very manual process. But this is something where I hope like LLMs may help, not may completely solve the problem, because you still need kind of, I think, humans to validate that it's actually a true term, but it could possibly suggest terms that can be inputted into those data warehouses, and probably maybe speed up the data curation process, right? And this is also very important in terms of for drugs to track the response of drugs as the patient, as the patient progresses with the disease, right? Sometimes you may not be able to determine, like collect data from a patient when they're responding or having a side effect from a drug. So you need to kind of capture routinely collected data from the hospitals, right? And then also a patient may get actually several samples of blood taken from them, as they're in a clinical trial or in a hospital. And so we can also find genomic markers that also are predictive of drug response here. Okay. So there's other kind of modalities of data. One that's kind of a lot of clinicians are talking about these days is wearables. So they see that as actually a really effective cost-cutting measure, because as I mentioned, one of the most expensive things for a clinical trial is actually getting the patient in to the clinic, right? It's even difficult to sometimes, patients have different schedules. They might not be able to come in for the clinicians to collect data, right? And also very expensive. The nurse or clinician might not be available. So can we actually collect data on the patient at home, right? Through wearable devices like smartwatches and so forth. So this is an early test, not done on patients. We actually tested this on healthcare workers during COVID, during the first wave of COVID. Lots of healthcare workers, they also got COVID, right? And then we tracked their symptoms using Apple Watches and compared it to questionnaires, so the kind of routine way of collecting symptoms. Okay? So here in this graph is like the red dot, the pink dots are like the visits, okay, where they actually collect their questionnaire symptom data, and then the lines here are where you collect the continuous longitudinal data from the wearable, the smartwatches. Okay, and then we can do kind of trajectory modeling to basically, we identified two different patterns of symptoms. Essentially, one pattern is basically long-term symptoms. So basically, long COVID is real, and that's been reported. And then there are cases where there's kind of short-term symptoms. Okay. So that's with kind of symptoms, kind of clinical data and symptoms. Also, you collect blood measures. You can collect blood measures from the patient, and so you can actually profile the DNA, RNA of those blood, right? So one type of marker, DNA marker that was described in the morning was epigenetic markers, DNA methylation. Right? So we did a study. I think Arthur is here. Actually did a study with on a Singapore cohort. These are healthy individuals, though, because we're still developing methodology for this, from kids where their methylation was profiled across different ages. Okay, so individual had different samples taken across their ages, and he fitted he developed a multi-mean GP to predict to fit on 3 previous time points of DNA methylation to predict a 4th time point. So using methylation data from ages of 3 months, 9 months, and 4 years, we can predict methylation sites and their status at 6 years. Okay. So why is that important? Because if you're trying to correlate a molecular marker like DNA methylation with a symptom, right? You may be able to collect maybe symptoms at 6 years, but you may not be able to collect their blood, right? Because again, patients may not want to come in to get their blood collected, but maybe you can collect some symptoms from, say, wearables, right? How do we benchmark whether we're actually predicting the right DNA methylation status? So what's known is that DNA methylation is actually very tightly associated with age. So actually, age of an individual. So we actually use the predicted methylation sites to estimate the age of the individual. So we know the age of the individual very precisely. So this is kind of the distribution from the GPs of the age estimate from the predicted methylation sites, and then we can see if we're closely predicting the actual age of the individual. Right? So otherwise, you're not really sure if you're really predicting if it's an unseen methylation site, whether you're predicting a true methylation value. Okay, so some general challenges, I think, with kind of machine learning with patient collected data. One is just accessing the data, right? Patient data has there's more and more regulation now in terms of, because I think from threats like cybersecurity threats and so forth, accessing the data is much more difficult. You're less likely to be able to actually download the data onto your computer or cluster of choice. You have to, a lot of times, access and analyze the data at the source, like at the hospitals and so forth, right? And then there's also having to go through ethics approvals. I think I probably spend half my time just on paperwork on ethics approvals these days. And then sometimes, like I mentioned, a lot of the data is actually unstructured. There's a lot of data processing that's needed to get into a machine readable state. And if you're lucky enough to kind of build models from patient data, interpretation is very important because the patient has an invested interest in that data, and the clinician needs to communicate back how the prediction was made, right? Because they're very critical decisions, right, for the clinician and the patient. So some future directions. So I think in this summer school, we talked a lot about kind of cellular and molecular level data. But there's kind of more interest in going like multi-scale, really going on to the level of tissues, organs, and the whole body, because it's not just that specific gene or cell or tissue that's affected, right? So there's some interest in taking some approaches used in manufacturing and the aerospace industry to kind of build essentially a digital model of a whole patient, okay? And that will need like kind of a combination of kind of machine learning and kind of more mechanistic modeling approaches, because that needs to make good predictions, but also needs to actually be interpretable. Okay, so this is still kind of somewhat of a dream. I think in terms of cardiovascular sciences, I think there's been really good models of the heart, right? So a lot of people have modeled kind of the whole organ of the heart, the dynamics and the fluid dynamics of the heart, and understanding kind of the omics profiles of the tissue in the heart, right? But I think there's still a challenge to expand to the whole human. The other side of things is that there's a lot of different sources of data now, right? It's not just from academia, right? There's industries collecting a lot of data, from clinical trials, pharmaceutical companies, a lot of technology companies are collecting data from patients even. So there's some partnerships for, say, things like psychiatric disorders, right? People have thought about using kind of social media data as a measure of kind of mental well-being as a kind of outcome in the trial, right? And then government entities, regulations really becoming important, and especially if AI algorithm is used as a diagnostic tool, it might be regulated, differently to some traditional diagnostic medical devices, right? So basically, it'll require this area of drug development will require basically pulling together data from a lot of different sources. Okay, so I'll just wrap up and thank my team in Sheffield and Singapore and some of my collaborators who I work on kind of drug development projects that I mentioned in this talk, and the funders. And finally, I'll say kind of self-serving promo, like there's openings in UK and Singapore in kind of this kind of drug development work. So yeah, email me if you're interested. But like I said, there's also a lot of players in industry and governments. Also, there's great opportunities there as well. Thanks.
This makes sense. Are there any questions?
Hi, thank you for your talk. It was really interesting. I was wondering specifically about the multi-output models. It seemed like the additional outputs you were looking at in addition to IC50 were kind of related to predicting individual dose responses, but I was wondering if you looked into any other outputs, especially with lead optimization, because you mentioned like specifically toxicity and binding affinity are correlated. So I don't know, is there any path towards multi-output models with lead optimization?
Yeah, that's a good suggestion. Yeah, I hadn't actually thought about that, but yeah, I guess that is why we're doing using multi-output models, right? Is that we could potentially incorporate these other measures of not just drug efficacy or response, right? But also like toxicity or bioavailability. I think the challenge is that it's hard to get those experimental data on the same samples. I haven't worked too much with kind of drug toxicity data, but they're not really done on a large variety of cell lines and animal models. I'm not sure exactly why, but I'm assuming it's some cost and just experimental setup. So it'll be a little bit hard to kind of associate the kind of genomic characteristics of the cells or the models with something like drug toxicity if they're only using just a very few models. I think that's kind of one challenge.
Thank you.
Thanks.
Other more questions?
Thanks very much. I actually liked the breadth of the different problems that you have been working with. And because of that, then here's a speculative question, because I'm particularly interested in understanding longevity. And when you were talking about high blood pressure, you were talking about a disease of exclusion. I think there's a lot of interest in augmenting health span at the moment, and so I was wondering, First, whether you have worked on understanding health span and longevity, and second, what would be the sort of, do you think that the UK Biobank and are there any other types of data sets that you think might be interesting should you, if you were thinking of working on longevity?
Yeah, thanks for that question. It's also my interest as well. So yeah, actually the division between kind of UK and Singapore groups is that the UK group focuses more on disease cohorts, and the Singapore cohort focuses a little bit on younger, middle-aged cohorts and even children to basically try to find kind of early markers of disease. So one actually motivation when we started the project with Arthur on the DNA methylation is because there's been some studies on older aging cohorts where they measured epigenetic age determined by DNA methylation and found that that was associated with mortality. So actually, just like whether they would accumulate other diseases later in life, right? So that's why we kind of wanted to see, how early can you go? Can you look even at children to see maybe there might be some early markers because the child develops and it is kind of like more influenced by environmental factors, and so yeah, so that's kind of one of the drivers there for that work. Your second question around like what data sets, so UK Biobank is one really rich data set. They're recruited as relatively healthy individuals, but it's longitudinal now. So they are a lot of those individuals are getting diseases, and UK Biobank is pulling in. I think they're working with NHS Digital to pull in health records, right? But it's not going to be enriched for disease, people with disease, right? There's another data set called 100,000 Genome Project that I worked on, and those are 100,000 NHS patients, so they're actually people with disease, either cancer, I think half of it is cancer, half of it non-cancer, and so they're really, it's genomic, whole genome data, plus a lot of health records from NHS Digital, and clinical records submitted by the physicians as part of the project. So that data set was locked down in a very inaccessible virtual machine for a long time. I think Magnus might have immense experience with it. They recently started using AWS, and you can actually run it on like AWS Workspace. And one of my research associates says it's a lot easier to work with now. So I think I might actually go back to that project. Like, I had a few projects that stalled, but I think that's still quite a rich data set to identify some disease signatures, disease predictors from the genome, right? And then you could potentially test that on a population cohort like the UK Biobank to see, examine what it means to be healthy, to have healthy aging or frailty.
Are there more questions? No more questions?
Feel ready to go home? Ready for your speech then?
So let's thank Dennis once again. With this lecture, we finished the summer school. Thanks everybody for coming. And if you have any feedback for us, we are very happy to receive it by email. We're planning to run again this school next year. So any feedback and things that we can improve will be much appreciated. And yeah.